Hello and welcome back to Educator.com. Today we're going to talk about translation. So as an overview, first thing we need to talk about is how we can link transcription to translation. After that, we will just give an overview of translation and go into it all the way through. So first we'll talk about ribosomes and their ribosomal subunits, and then we'll go through each part of translation, so starting with the charging of tRNA, all the way through to initiation, elongation, and termination of translation. And finally, we'll briefly talk about the consequences of altering the genetic code. So transcription, once again, to remind us from last time, is making RNA from DNA. Okay, so we synthesize RNA, okay, complementary to DNA. All right, and this is going to occur in the nucleus. All right, this is important to remember. Okay. Now, translation is the process of synthesizing a polypeptide from an mRNA transcript. All right, so synthesize polypeptide from mRNA transcript. Now, if we have our tRNAs, our rRNAs, those aren't going to be made into proteins, so you aren't going to translate those RNAs. All right, and obviously our non-coding RNAs you won't translate either. But our mRNA transcripts are the ones that are going to go through translation very often. All right, so we have our mRNA being translated in a 5' to 3' direction, all right, just as if we were talking about DNA synthesis happening in a 5' to 3', meaning the next base is added to the 3' end. Okay, so mRNA is translated 5' to 3', okay, 3 bases at a time. All right, this is important to know. All right, 3 bases at a time, which we call a codon. All right. So each 3 base pair unit, or each 3 base unit on an mRNA is called a codon, and each one of those codons will code for a specific amino acid or a stop. All right? So the mRNA gets translated 3 bases at a time by base pairing with a tRNA, okay, having a complementary anticodon. So your codon and your anticodon will match up via a base pair. So an example being, let's say, UAG on your codon, your mRNA, okay, would match up with AUC on your tRNA. All right, and remember, we would have the hydrogen bonds between there, right? Okay. Now, translation is going to occur in the cytoplasm. And it will either occur inside ribosomes that are located on the rough endoplasmic reticulum, okay, which the endoplasmic reticulum is studded with ribosomes. That's why it's called the rough endoplasmic reticulum. We also have the smooth ER. So it's going to be either in the rough ER or in the cytosol itself. All right. And so if we look at this depiction down here, we can see that here is our nucleus. All right. We have transcription occurring in the nucleus, so DNA being made into RNA. In this case, we're looking at mRNA. That mRNA can leave the nucleus into the cytoplasm, at which point right here, this is our ribosome. Okay, and it's going in this direction. Our ribosome can be the site of protein synthesis by reading the mRNA 1 codon at a time and recruiting our tRNAs to come in and add its individual amino acid to the growing polypeptide chain shown here. Okay, and we'll go over each one of these steps of translation today, and hopefully we'll have a very good understanding by the end of this unit. So, as an overview, what's happening in translation? All right, so first things first. We have a ribosome binding to an mRNA molecule. All right, it's going to bind to it and it's going to search for the start codon. Okay, now a start codon, this is going to be found once again on the mRNA. Your start codon is going to be AUG. And remember, this is on the mRNA. Okay, this AUG will code for methionine. Okay, methionine. Now, inside the ribosome, charged tRNAs, so that means they're carrying an amino acid, will base pair to the mRNA via the complementarity found between the anticodon on the tRNA and the codon on the mRNA. Okay. So this is important. Codon on the mRNA, anticodon on the tRNA. Okay. Now the next step is we have the amino acids being transferred from the tRNA and being linked to the growing polypeptide chain via a peptide bond, otherwise known as an amide bond. All right? And this growing polypeptide is still being associated with the ribosome itself. Okay. Now, spent tRNAs, so tRNAs that no longer have an amino acid because it's already given its amino acid and let it attach to the growing polypeptide chain, these spent tRNAs get released from the ribosome via the E site, okay, or the exit site, and we'll talk about that later. And then the process of bringing in a new tRNA, having a peptide bond being synthesized and a growing polypeptide continuing in its synthesis will continue until you reach a stop codon. And once again, remember it's a codon, so it'll be found on the mRNA. And our stop codons are UAA, UGA, and UAG. Okay, so we continue till we reach a stop codon, and then what will happen is instead of a tRNA coming in, we have a protein called a release factor coming in, and what that does is it basically hydrolyzes or cleaves the polypeptide, releasing it from the ribosome and allowing it to fold into its final tertiary shape. Okay. So those are our main steps. That's our overview.